All right, I have 1 o'clock on my watch, so I'm going to go ahead and get us started today. We still have some folks filing in, but we're going to start so that we can maximize most of this hour. So thanks everyone for joining us today. I hope it's sunny and beautiful where you are. It's rainy and cloudy where I am. But today we're going to talk about the impact of pesticides on the microbiome and health of honeybees. We have a great speaker, Dr. Kasie Raymann, waiting here to chat with us today. But before we start, I'm just going to go over a few basic rules that I think everybody has seen over the last year and a half. So microphones were muted upon entry. If you unmuted your microphone, go ahead and please mute it again. And also, if you would, turn off your camera to minimize any additional distraction. There'll be plenty of time for questions after Dr. Raymann's presentation has ended. If you do have a question throughout the presentation, please feel free to drop it in the chat. I'll be keeping an eye on the chat so that I can read out those questions afterwards. Dr. Raymann's going to provide her contact information at the end of the presentation. So if you want to connect with her afterwards, you can. And then this webinar is recording currently, and it's going to be available on our YouTube channel in the next few days. And I will send out an email through Eventbrite with the link to let everybody know when the recording is available. So just a really quick background on our group. We are the North Carolina Pollinator Conservation Alliance, and we are loosely a partnership of 27 different agencies and organizations across the state. We all have a common interest in pollinator outreach and conservation. And within our partnership, we have 6 committees: plant resources, outreach, habitat assessment, research, energy, and pesticide stewardship. And this webinar series is being brought to you by our outreach committee. And this, incidentally, is the last in our fall series. We had 2 other webinars earlier this fall, and they're also on our YouTube channel if you missed them and want to check them out. And if you want to learn more about our group, please check out our website, ncpollinatoralliance.org. We are also on social media, including Twitter and Facebook. And we are so successful because of all of our partners. We would not be where we are or have the information that we are lucky to have without so many great partners. So I'm going to stop sharing my screen and let Dr. Raymann load her presentation. And in the meantime, I'm going to tell you a little bit about her. Dr. Kasie Raymann is a microbial ecologist who uses the honeybee as a model system to study the evolution and dynamics of host-associated microbial communities. She obtained her Bachelor of Science in Biology from Indiana University in 2011 and her PhD in Microbiology and Evolutionary Genomics from Pasteur Institute in Paris, France in 2014. Dr. Raymann was a postdoctoral research fellow at the University of Texas at Austin from 2014 to 2017 under the supervision of Nancy Moran. In 2018, she became an assistant professor in the biology department at the University of North Carolina at Greensboro, where she is also one of the directors of the UNCG Plant and Pollinator Center. Her previous research has investigated the impacts of chemical perturbation, for example, antibiotics and pesticides, on the honeybee gut microbiome and honeybee health. From this work, Dr. Raymann has shown that both antibiotic and pesticide exposure can severely alter the gut microbiome of honeybees, which leads to increased pathogen infection and mortality. So I'm going to go ahead and hand it off to Dr. Raymann so she can tell us more.
Sorry, I was muted and I couldn't figure out how to get unmuted. All right, thank you so much for inviting me to give this webinar. I'm excited to speak to all of you. And I think that Gabrielle basically told you everything about me, so I don't need to say anything else. No, I'm kidding. So today I'm going to specifically talk about pesticides and how they impact the microbiome and health of honeybees. And I'll be focusing on glyphosate because I was told that this is a lot of interest to all of you right now. So Gabrielle already kind of told you about my history and how I got to where I am now at UNCG. But what I wanted to point out is that everything that I'm going to speak to you about today is work that I did as a postdoc when I was at the University of Texas. Some of the work was done by myself Eric, which was a PhD student at the time when I was there. And then of course, the head of the lab, the PI, Nancy Moran. So I just want to give them credit. And then a lot of the work I'm going to talk about today was actually done by Eric after I left the lab. So just to let you know a little bit about microbes and how important they are. So life on Earth is actually sustained by microbes. And these microbes can include everything from bacteria, archaea, fungi, and as well as viruses. And microbes are literally everywhere. They can be found at the very bottom of the ocean, to on animals, within animals, and even in the air we breathe. And although you might think that microbes are bad, of course, especially right now with the COVID pandemic, they are really important for a lot of things. They're important for our environment. They are involved in a lot of chemical cycling and nutrient cycling within the environment. They are essential for plant growth and productivity. We use them for a lot of applied microbial processes like making biofuels, making a lot of foods that we love to eat. And for the purpose of this talk, I want to focus on the fact that they're really important for animal health, nutrition, and development. So currently in my lab and what I've been doing in the last few years before I started my lab is that we focus on understanding the evolution, function, and importance of honeybee-associated microbes. And when I say honeybee-associated microbes throughout this talk, I'm going to be referring to that as the microbiome. So just a little bit of background about microbes and honeybees. So there's been a really extensive amount of research that's been done over about the past decade. And you might have noticed that not only in honeybees, but this work has been done in all animals, including humans, really showing how important microbes are to animal health and fitness. But in honeybees in particular, this research has revealed that honeybees have a very highly conserved microbiome, and it's socially transmitted. And I'm going to talk a little bit more in detail about that in a minute. Their gut microbiome is composed of host specialized bacterial species. And when I say that, I mean the species that we find, the bacterial species that we find within the gut are restricted to living inside of honeybees, and we don't find them environmentally. So they're really specific to the honeybee microbiome. Their gut microbiome is highly structured. And again, I'm going to come back to that in just a little bit. And what's the most important thing that we've really realized over through all this research is that their microbiome plays a very important role in their health. So I want to talk about how honeybees acquire their microbiome, but first I want to give you a little background about the hive makeup. I know a lot of you probably already know this, but just to talk about why we mostly focus on workers. So workers are going to make up 85 to 95% of the hive, depending on the time of the year. They perform all the tasks in the hive, and they live between about 1 to 3 months, depending on the season. We have drones that can either not be present in the hive, or they can make up about 15% of the hive. They're taken care of by workers, and they live about 1 to 2 months. And then, of course, in the hive, we have only one queen, and she's also taken care of by the workers. And she should be living for about 2 to 6 years. I'm sure you all know that's not really happening right now, but that's a whole nother story that we have to talk about later. So I wanted to highlight this because most of the work that's being done on honeybees is going to focus on workers. And a lot of the things that we know about the honeybee microbiome in particular is about workers. We do know the composition of the microbiome of drones and queens. They contain some of the same microbes of workers, but they do have slightly different microbiomes. So in terms of workers, and we assume this is also how drones and queens acquire their microbiome. So basically what happens is that as their larvae, they're fed by workers. So they're getting some bacteria. So on this y-axis here, you have the total number of bacteria that are going to be present. So they're getting some microbes in this stage, but the composition is really erratic. Then when they pupate, they shed their gut lining, and then they lose all of the microbial composition, and they only have a few numbers of bacteria. And then once they emerge as adults, this is when they're actually going to acquire their microbiome, but that takes a few days. There's still this sort of erratic composition. And then after day 4, they generally establish this characteristic microbiome that I'm going to tell you a lot more about in a minute. So I told you that honeybees acquire their microbiome vertically or socially within the hive. And so there's been some studies done to show exactly the importance of different routes of acquisition. And so by far the most important way that honeybees acquire their microbiome after they emerge is through fecal oral transmission. They get some of their microbiome from hive material, and then they also get some of their microbiomes from oral trophallaxis. So now a little bit more about the structure of the honeybee microbiome. So there are 8 major bacterial species. So here that's shown, and I'm showing you the major species here. You don't need to focus on the names, but these 8 that are shown here make up about 95% of the microbiome composition of a honeybee. And then you only have a very small, between like 1 to 5% of these other, and they're typically like environmentally acquired microbes that they get as they're foraging and things like that. What's really important to know is that there are actually 5 bacterial species, which we call the core, and I'll call these the core throughout the talk, because these species are found in every single honeybee around the world. So no matter where you sample a honeybee from, you're always going to find these 5 species present in their gut, and that indicates to us that these are very, very important. Also, we can find several of these core species present in other related bee species, including bumblebees. And so it's predicted that these specific bee gut bacteria have actually co-evolved with their hosts for millions of years, making them have this very tight relationship with each other. I also mentioned that the gut was very highly structured, and so I just want to go over a little bit about the composition throughout the gut microbiome. And so here we have the mouth, and then the crop, or the honey crop, as some might call it. And here you have very few bacteria, and when you do find bacteria, they're mostly associated with nectar and things that they pick up in the environment. Then we have the midgut, and there are very few bacteria here because they don't have a stable lining. This lining is shed quite often throughout their lifetime. And then once we get to the hindgut, that's where we're going to see the majority of the microbes inhabiting. And these are going to be the stable areas. And so you have the pylorus, where you have this local colonization by one particular microbe. And then in the ileum, which would kind of be like our small intestine, if you want to think of it that way, this is dominated by 2 particular species, one called Snodgrassella and one called Gilliamella. And what's really interesting here is that Snodgrassella forms a biofilm along the epithelial lining of the ileum, and then Gilliamella forms a biofilm on top of that. So this is where we have this really tight structure and this really big association between these 2 important core species. Then when we move into the rectum, you have even higher number of bacterial cells. And this is where we're dominated by things like Lactobacillus and Bifidobacterium, which are gram-positives, that are also found in humans and other animals and are thought to be very beneficial microbes. But the structure in this area is a lot less defined. You kind of just have a hodgepodge of the microbes living around in this area. So there are several factors that can alter the gut microbiome. But first, I just wanted to point out to you that, through a lot of research that's been done in the past few years, we've been able to show that the honeybee microbiome does play a large role in immune function, metabolism, and this is really interesting. So not only in metabolism of sugars that they generally eat and plant material, but also of toxic sugars that they otherwise would not be able to ingest if they didn't have the microbes present. It also plays a role in their hormones and their behavior, their growth and development, as well as the susceptibility to pathogens. So there are several factors that have been shown scientifically to influence microbiome composition, and these include several things like diet, climate and season, the age and the caste. As I told you, queens, drones, and workers are a little bit different. And of course, exposure to chemicals like antibiotics and pesticides and other toxic molecules. And so when exposed to these different things, it can alter the microbiome, and then this can have cascading effects impacting their immune function, their metabolism, and as I said, other things like development and pathogen susceptibility. So here for this talk, we're going to focus on pesticides. And so I just wanted to go over a little bit about how honeybees can be exposed to pesticides. So there's several different things. Obviously, direct contact if pesticides are being sprayed while the bees are out foraging. There can also be drift that occurs. So you can have spray drift or like seed coat drift that moves to different areas and the bees get exposed to it. There have been many studies showing that pesticides can be found in nectar and pollen, as well as in the water sources, as bees do drink water sometimes. And then many studies showing that the bees bring back these residues and then they can be stored in the hive materials. For example, they can be found in honey, the stored pollen, as well as in the wax in the hive. So today we're going to focus mostly or exclusively on glyphosate. So I wanted to give a little bit of background about that. So glyphosate is the active ingredient in Roundup. It's the most commonly used weed killer in the United States. And in fact, the use of glyphosate or Roundup has increased significantly over the past 2 decades. I couldn't find anything more recent than 2016, but just looking from 1996 to 2016, you can see the darker areas are meaning that you're having increased pesticide use in those areas. And you can see that that's happening a lot in the Midwest, but also includes on the East Coast and in North Carolina. So because of all of this excessive use, of course, non-target organisms, for example, honeybees, are going to be exposed to glyphosate in the environment. And the concentration that's typically applied in crop and non-crop areas is larger than 30 micromolar. And this is important for me to bring up because I just want to point out that all the studies that I'm going to show you today were done using a far less concentration than this. So just a little bit about the mechanism of glyphosate. So glyphosate is going to impact what's called the shikimate pathway. And the shikimate pathway is actually a metabolic pathway that's present in all plants, and it's also present in most bacteria. And this pathway is responsible for producing essential amino acids that are needed for growth and survival of an organism. And so glyphosate, what it does is that it blocks the shikimate pathway, and then it prevents these essential amino acids from being formed. And so then that would eventually lead to the organism stopping growth and then dying. A little bit more about the mechanism of glyphosate. So it does inhibit the 6th enzyme in the shikimate pathway. And this enzyme has a really long name, so we're just going to give you the abbreviation. It's called EPS. And there are 2 different classes of this enzyme. So there's class 1, and this is the class that's present in all plants, and it's present in most bacteria. And then we have class 2, and this is present in some bacteria. So as you can see, we know that glyphosate doesn't directly target bees, so it only targets plants and bacteria. But what about the honeybee gut bacteria? And so that's really what we wanted to know. So the first thing we did was try to look at the genomes of these microbes, so their genomic content, and see if we could identify whether or not they had the target for glyphosate. And what we found is that actually 4 out of the 5 core bacteria contain the glyphosate target. 3 of these have the class 1, so that meaning that they're very susceptible to glyphosate. And then 1 has the class 2, so it's tolerant. And then 1 of the core doesn't have the target at all. So this gave us the inclination that glyphosate could be severely disrupting, especially these susceptible microbes that are really important to honeybees. So the first thing, of course, we wanted to do is just see if we could confirm that glyphosate would actually inhibit or stop the growth of these susceptible bacteria. So we took these isolates of these bacteria when we grow them in the lab in the presence of glyphosate. And what we found, and I'm just showing you an example of 2 strains we tested, so the Snogracella and Gilliamella. And what we found here is that this is the control growth where they weren't exposed to any glyphosate. And then here's the growth when they were exposed to glyphosate. And so you can see that their growth was inhibited when we put them in the presence of it. So we've confirmed this in the lab now, but now does this actually occur within the honeybee? It's a good question because we don't know how much concentration is going to actually reach the gut of the bee, or if it's going to affect the microbes in the real system not just in the lab. So what we did for this is that we exposed bees in the lab to 0.1 millimolar of glyphosate. So remember, I told you 30 millimolar is how much it's actively sprayed in the field. And then we had controls that we only gave sugar syrup to, and we characterized their microbiome composition after exposure. And what we found was that there were 6-fold fewer total bacterial cells in honeybees that were exposed to glyphosate. And we also found a significant decrease in this core bacteria, Snodgrassella alvi, in bees that were given the glyphosate treatment. So this gave us the indication that glyphosate given at a low dose can actually have a big impact on the composition of the microbiome. So then we wanted to know if this is having any impact on the health of honeybees. And so to do this, we tested if glyphosate is increasing the susceptibility to pathogen infection in bees. I just have to give you a little bit of context here. So we used this opportunistic bacterial pathogen Serratia marcescens, which I did a lot of work on and have shown that it is a pathogen of honeybees, but it really only causes disease in compromised bees. So this was a good microbe for us to use, or pathogen for us to use, because that means that it shouldn't really have much effect on bees that don't have any disruption of their microbiome or immune compromisation, but it should impact them if they are compromised. So we took young bees, we exposed them to glyphosate, and then we exposed them to this pathogen Serratia marcescens. And what we found was that bees that were exposed to glyphosate showed significantly increased mortality. So that would be down here on the bottom line after pathogen infection. So the pathogen is at quite high doses. It does kill some of the bees that weren't treated, but you can see that if they are given glyphosate, that this goes down, and almost 100% of the bees ended up dying after 8 days. So all of this work was done by Eric, me, and Nancy, and it was published in 2018. So if you're interested in reading more about that paper, you can find it here. If you can't get access to it, let me know, and I'm happy to send it to you. So next, a little bit more about Roundup. So we know glyphosate is the main active ingredient. However, there's many other unknown ingredients in Roundup. So this is just one version of Roundup. There's lots of different versions that have different concentrations, but overall, you're going to see the glyphosate's the main active ingredient, and can range from 18 to 50% if you're using a really concentrated version. But then you have this thing, other ingredients, that make up a large proportion, and sometimes 80% or more of the concentration of whatever is in Roundup. We don't know what these ingredients are. So after I left the lab, Eric was finishing his PhD, and he continued to work on this topic, but this time he really wanted to look at how Roundup impacts the honeybee microbiome and do not only lab experiments, which is what we did together, but also do some field experiments. And so for the rest of the talk, I'm going to talk about this paper that was published in 2020 by Eric and other co-authors in Nancy's lab. And this is just a really impressive amount of work. So there's a lot to show you, and it's extremely well done. And I have some exciting things to tell you about from it. So first of all, glyphosate versus Roundup. So Eric exposed bees in the lab to 1 millimolar of glyphosate, and Roundup, that the concentration he gave also had 1 millimolar of glyphosate, and then of course controls. So here we have bees that he captured. He then exposed them in the lab, and then he sampled them after 6 days. And here are the different treatment groups we have. And what he found is that glyphosate and Roundup both impacted the microbiome composition. And so I'm always keeping this here kind of as a reference for you to see those core bacteria. So we see that there's a decrease in overall bacteria, both in the glyphosate and Roundup treated bees in comparison to controls. We look at Snodgrassella, one of our cores here, and we see that the abundance of this bacteria is significantly decreased in both glyphosate and Roundup. For Gilliamella, another core species, we see that it's significantly decreased only in Roundup and not so much in the glyphosate. And then the same here for Bifidobacterium. We see a larger impact of Roundup on this bacteria, and it's significantly decreasing the abundance. This over here is a way that we look at the overall composition of the microbiome and compare it against the different treatment groups. So in gray here, you have the control, in green, you have the glyphosate, and then in orange, you have the Roundup. And basically what this shows is if you have clustering together, that means they're similar. And the further away they get, that means the less similar they are. And so you can see that the glyphosate and Roundup treatment made the bees less similar in microbiome composition than controls, and that it impacted them in different ways because they're not completely overlapping. And then overall, what I wanted you to take away from this is that we found, or they found, that Roundup had a larger impact on the gut microbiome than glyphosate alone, which is interesting, and we'll kind of revisit this in a little bit. So then he wanted to know if the microbiome can actually recover after exposure. So he exposed bees to 0.1% of Roundup. So basically what happened is that he captured bees, he exposed them in the lab, then he sampled some of those bees, then he released the bees back into the hive, and then he sampled them again at different time points. So at day 3 and day 5 post recovery. So what he found from this was similar to what was shown before, is that Roundup impacted the microbiome significantly at all days post-treatment. And so at day 3 and 5, there were significant drops in the total number of bacteria, Snodgrassella, Gilliamella, Bifidobacterium, and then the only outlier, and then a little bit in Lactobacillus firm 5, which doesn't have the target for glyphosate, but because of the shifts in abundance of other microbes, it could have an indirect effect on some of the other community members. So then he wanted to look at how glyphosate and Roundup, or Roundup in general, impacts in hive survival. So what was done here is that bees were captured, as I explained before. They were brought into the lab and exposed, and then they were marked. So you could mark the control and the treatment groups with different colored dots on their back, re-release them into the hive, and then go back and sample and see if you can find those bees again after a certain period of time, which in this instance is going to be 3 days. So 3 days after he had released them back into the hive, he went out and he counted how many bees he could find. And what he found was that only 27% of the bees that were exposed to the 1.1% Roundup were recovered. And this was actually 20% less than how many control bees he was able to recover from the hive. So this suggests that Roundup is actually reducing the survival rate within the hive. So next, since everything had been done kind of giving bees sugar syrup in the lab and then returning them to the hive, he wanted to look at how it would impact them if they were only exposed within the hive. So bees were treated in the hive with 0.1% of Roundup, and then of course controls were just given sugar syrup. And so here was the experimental design. They had a bunch of different hives, one that was control, one that was treated, and they were given treatments in sugar syrup. So they had Roundup in those sugar syrup, or in controls they didn't. And then they sampled bees at weeks 0, 1, 3, 4, and 7. And they also sampled honey, and we'll talk about that in a minute. And they also took bees at week 4 and tested them for pathogen susceptibility. So in terms of microbiome composition, what they found again was that there was a reduction in all bacteria when you looked at control versus the treatment hives at all time points, a reduction in our core species Snodgrassella at time points 1, 3, 4, and 7 weeks. and then a reduction in bifidobacteria compared to controls at weeks 3, 4, and 7. So overall, this just shows us that the microbiome was disrupted in bees that were treated in the hive, and this continued to occur over multiple time points. So then they looked at pathogen susceptibility. And so again, using this Serratia challenge that I described to you earlier. So what they found here is that when they sampled bees on week 4 that had been treated in the hive, they found again that bees that had been given the 0.1% of Roundup and exposed to Serratia showed significantly more death than control bees that were given just Roundup alone, or control bees that were also exposed to Serratia. So again, this suggests that exposure to Roundup is increasing susceptibility to this opportunistic pathogen. Next, they looked at honey contamination. And so they did another experiment where they used 2 different concentrations of Roundup. So really, really low, so 0.001%, and then a 0.1% as they'd been using before. They again treated the hives in sugar syrup, and then they sampled the honey after week 4. And they used a mass spec to quantify how much glyphosate was actually present in the honey, the stored honey in the hive. So what they found here is that we see this with the triangle dots, that is the highest percent, so the 0.1% of Roundup, and the little square orange circles here, or orange squares here, are the lower level. And they found that there were very high levels of glyphosate in the 0.1% treatment hives, going up to about 1,500 micrograms per milliliter. And this seems to be increasing over time. And although you couldn't really see the difference here because of the scale and how much accumulated within the higher concentration group, if we zoom in just on this alone, you can see that even at 0.001%, there's a significantly increased amount of glyphosate concentration present in honey, even at this really low level. The next thing that they were interested in testing is how topical exposure would impact survival rate. So in the worst case scenario, you know, bees could be out foraging when glyphosate is sprayed or Roundup is sprayed. And so they wanted to look at how this could affect survival. So what they did here is that they topically exposed bees to different concentrations of Roundup. So they captured bees from the hive, brought them into the lab. They sprayed them with a mixture of different concentrations of Roundup, and then they monitored survival for 24 hours after that. And what they found was that concentrations of Roundup at 0.5 up to 3% significantly decreased survival in honeybees, as you can see here. And this started in the highest concentrations even 6 hours after they were topically exposed. And what I want to mention here, I'm not showing the graph for it, but they also did test whether glyphosate alone would cause the same effect. And they did not see any difference in death between the control and the groups that were treated with these percentages of glyphosate. So this is super interesting because it really suggests that it's these other components that are present in Roundup that are causing this mortality within bees. They did another experiment with topical exposure, and this time it was to look at whether or not topical exposure and then re-release to the hive would result in decreased survivalship within the hive, not just in the lab. So they again brought them in, gave them a topical exposure, released them, and then 3 days after they went back and they had marked those bees. So they again counted how many bees from each group that they could find. They also evaluated bees that they sampled to see how these different concentrations impacted the microbiome composition. And so here are the results from this. So for each of these, you have Roundup concentration, so it's increasing along the x-axis, and then the number of bees they were able to recover after 3 days. So you can see the control, they could recapture about 50%, and then the number of bees that they could find in the hive after treatment decreased as the concentration of Roundup increased. So this also fits with the results I showed you on the last slide, but suggests that these results are really dose dependent. Also, to have a proxy for how it impacts the microbiome, they took Snodgrassella. So if you remember, it's that species that's been affected the most in all the other studies. And they looked at the total number of Snodgrassella they could find within the microbiome at different concentrations. And they found that at concentrations of 1 to 3% of Roundup, it significantly reduced the number of this bacteria within the microbiome. So overall, these results really show that Roundup reduces survivalship, both in the lab and in the hive, but that it also can affect the gut microbiome. So together, now we see that oral exposure as well as topical exposure can have a big effect on these metrics. So aside from the microbiome, there's been a lot of other studies looking at indirect effects of glyphosate, and I just am going to mention a few. There's been tons of studies, so I'm sure I'm probably missing some, but these are not related to the microbiome, mostly related to behavioral effects on bees. And one thing that's really interesting is that it's been shown that foraging bees actually prefer food sources that have low concentrations of glyphosate, which is a little scary. So that means that they're probably going to those sources and then getting exposed more and bringing them back to the hive. It's been shown that there was an association between an odor and the sucrose reward. So when the bees smell something on a plant, there's a specific odor that tells them that this is a really good food source. And apparently this has been shown to be impaired by acute exposure to glyphosate. Glyphosate also has been shown to diminish the short-term memory retention of honeybees, to alter their homeward flights. So it changes the way that they come back from foraging, and sometimes can cause them to actually get lost and not ever return to the hive. There's also been a study that showed that it negatively affects larval development. So bees that were exposed to glyphosate, they took longer to develop, they were smaller, and a lot of them didn't survive through pupation. And it's been shown that it reduces sucrose responsiveness. So in tests where they give bees different levels of sugar and see how much they respond to it, it seems like if they're exposed to glyphosate that they are less responsive to different concentrations. And then in this study as well, they showed that it significantly affects olfactory learning. So again, that would have something to do a little bit with short-term memory retention, and that it negatively affects memory ability, and that it can decrease climbing ability in honeybees. So these, like I said, there's been tons of studies done on this stuff. Of course, it's not my area of expertise, but it gives us the idea that it doesn't only have impacts on the microbiome, but it can have severe impacts on honeybee behavior. So takeaway messages: glyphosate and Roundup exposure can alter the gut microbiome, and then this can have major impacts on bee health. Both glyphosate and Roundup can increase susceptibility to opportunistic pathogen. Topical exposure to Roundup can alter the microbiome, and it also reduces survival both in the lab and in the hive. And that the other ingredients in Roundup are as, or maybe possibly more dangerous than glyphosate alone. And then, of course, glyphosate has been shown to greatly impact many important honeybee behaviors and development, like I just talked about on the last slide. And that they both should be used with caution and in limitation. Of course, they're necessary. Sometimes it's necessary to use, weed killers and herbicides, but given this information, unless it's absolutely necessary, I would say it's best to avoid it. So I didn't get to talk about anything I'm really doing in my lab right now because I wanted to give you all the information about glyphosate and honeybees. But I just wanted to give a brief overview about some of the applied honeybee projects that we're doing in my lab, in case you want to follow up with me or ask me questions after. So one big project we're doing is that we're looking at how in-hive treatments impact the reproductive health and the microbiomes of queens and drones. We're also determining what factors are shaping the fine scale community composition of honeybees. We are testing how efficient commercial bee probiotics are on restoring the honeybee gut microbiome after antibiotic treatment. And there's a little project we've been doing where we're looking at what pollinator marketed flowers are more attractive to honeybees versus native pollinators or both. So with that, I want to thank you all for listening. Of course, I definitely have to give a big shout out to Eric and Nancy, who did the bulk of this work, also with their collaborators that worked on the paper with them. The work's been funded by the USDA, NIH. Eric's PhD was funded by several different entities in Brazil. And then the University of Texas at Austin, there were the Department of Integrative Biology is where the work was done. And then Eric received a fellowship. And then even though I didn't get to talk about my lab much, I just wanted to give a shout out and a thanks to my lab for all their hard work and support. And here's a couple pictures of us pre-pandemic. Hopefully we'll get some new lab photos soon. So with that, I'm happy to take any questions you have. And here's all my contact information if you want to follow up with me later. And thank you all for listening.
All right, thank you, Kasie. That was very interesting and relevant. And we have a lot of questions, so I'm just going to jump straight into those.
Do you think I can stop sharing, or do you want me to keep that?
No, maybe just keep up that slide for a minute or two so folks can have the opportunity to write down your contact information.
Okay.
And then you can stop sharing.
Perfect.
So the first question is, what is your opinion of the new products such as Super DFM? Is there evidence that they are beneficial or necessary?
So I just mentioned that we are testing that. So I have pretty strong opinions about it, honestly. So Super DFM, if you look at the label, it's composed of nothing that even colonizes the honeybee gut microbiome. They made it out of microbes from the human microbiome, from other animals. There's even like baker's yeast in there. So I think it's quite a scam. So that's what we've been testing. So we have a bunch of hives that we've worked with a master beekeeper here in North Carolina, Burton Beasley. I'm sure some of you know him, where we treated a bunch of hives with terramycin, and then we gave different probiotic treatments after. We have preliminary results on that already. And what we find is that no, it has no effect on restoring the microbiome. And we have never identified any of the microbes that are in those cocktails in a single bee. So they're not colonizing the bee, and they're definitely not restoring the microbiome after treatment. But more to come on that.
So Linda Burns had a question that she posed about halfway through your presentation. Are these bees nurse bees or foragers, or is that known? So Linda, if you want to unmute yourself just for a moment and clarify, because I think that was probably more applicable to that point in the presentation.
Sure, thanks. This was during the portion where the researcher was feeding the syrup and then releasing the bees back into the hive and then trying to go back in and then recover those bees. And so I was wondering, or if those bees were nurse bees and they would not be foraging and they'd be staying in the hive, versus they were forager bees and so they went out and they never came back in, with regard to how many he could then locate. Does that make sense?
Yeah. And I've done this experiment before with antibiotics and other things. It's actually just the problem with capturing the control bees. Even so, the reason that the level of control bees we can recapture is so low is because to be honest, it takes hours to go through the hive, through all the boxes and try to pull out as many of the painted bees as you can. And yes, because they're not age controlled, some of them are out foraging, but they're randomized when they're sampled. So you would expect to, try at least be able to capture around the same amount, because the same amount of them should have transitioned to foraging for control and treatment, if that makes sense.
Yes, I understand. Thank you.
Okay, the next question is from Phyllis. Do you know if any of our native bees have the same or similar core bacteria? Do you know of any similar studies with other bee species?
No, unfortunately, I think there are maybe a couple on bumblebees, and yes, bumblebees have very similar microbiomes to honeybees. The only difference is that their microbiome isn't as stable. And the reason for that is that they have a bottleneck every winter. So the queen, goes on her own. She doesn't keep the whole colony, and then she restarts the colony every year. So you don't have as conserved of a microbiome in bumblebees, but they do have the same core species present most of the time. And so I would predict that you would have the same results with bumblebees. In terms of like solitary bees, they haven't been studied a lot, but in general, what has been shown is that they don't have a conserved microbiome at all, and it's not really similar to social bees. So that's a big thing here that we have to realize, is that social bees have this conserved important microbiome because they're social and because they can acquire it directly from, social interactions within the hives, whereas solitary bees don't have that. That doesn't mean that exposure wouldn't have any negative impacts on solitary bees, but nobody's looked at it, and it would be a little bit harder to characterize how it's impacting it, considering that they don't have this like very conserved microbiome that we could say, oh, that's exactly how it's being impacted.
Kasie, do you know, because I was also curious about the native bee aspect of it, do you know if there are any efforts to create studies to look at that in the near future?
I hope so. I know that, I'm not sure if like Quinn McFrederick is doing anything with pesticides. He does a lot of work on the microbiome of native bees, mostly, bumblebees, and so maybe that would be something that he might be thinking about doing. I'm not sure if anyone in Nancy's lab is working on that either. I think it's really important, but of course, everybody ends up focusing on honeybees in the end because they're so agriculturally important, and we kind of like put to the wayside the poor native pollinators that are just as important. But it's something to think about, and I definitely think if nobody's looking into it, it would be something I would be interested in looking at in the future.
Yeah, definitely. This next question is a two-part question, so I'm going to read the first part. Has the impact of pesticides, including glyphosate, on the bees' cognitive behavior and learning abilities been investigated?
What was the first part? Has what?
Has the impact of pesticides on bees' cognitive behavior and learning abilities also been investigated?
Yeah, so that was the last little brief slide I showed. There's been actually a lot of work done looking at pesticides and how they impact behavior, and glyphosate in general, has been shown to impair memory and retention of memory and behavior as well. Like if you think about behavior in terms of like reacting to different sucrose contents and being able to climb well. So yeah, for that, but and for other pesticides, like for example, imidacloprid, which is, an insecticide that has even more severe impacts on honeybee behavior and cognitive ability. So, and that seems more reasonable to think about because it does directly affect insects' neural pathways. But with glyphosate, yeah, there is impacts on that, but we're not really sure how it's causing those impacts since it doesn't target directly the bee themselves, whether it be because of the disruption of the microbiome, or there's some stuff I didn't show you that's unpublished by Eric as well, showing that glyphosate causes reduction in immune response in bees. So it could be that the cognitive and behavioral things are kind of an indirect effect of glyphosate, but they're definitely there.
The second part of that question is, you mentioned a diverse group of bacteria that were differently affected by glyphosate. What is the reason behind this difference in getting affected?
So if you remember, I told you that we looked at each of the microbes that are present in the bee, and we identified whether or not they had the target for glyphosate. And we found that some of them do have the target, and that they have the susceptible target. So that means that if they're exposed to it, their growth is going to be inhibited. Some of them have the tolerant target, which means that they can be, but they have more tolerance to it. And then we have some member, well, one major member that doesn't have the target at all, so it shouldn't be affected. And the only way that it would be affected is just that the other members in the community are affected. And so that has cascading effects on the overall composition, because if you perturb one species, that perturbs the community and their interactions together. And so then that can even cause effects on things that don't have the target for glyphosate. I don't know if I answered the question right, but.
I think so. I think that was a good answer to that question. The next question, do the inactive ingredients in Roundup include chemicals like surfactants to allow the product to stick to and be taken up by the leaves? And how would those affect bee survival?
I honestly have zero idea what the other ingredients are. I don't know if you can find that out. Honestly, I think, Bayer, Monsanto, or whoever's making it now doesn't want anybody to know what they are because it's like a patent. But what Eric clearly showed is that Roundup has a much bigger effect on bees than glyphosate alone. So there has to be something in those other ingredients that are actually probably more detrimental than glyphosate itself, which, kind of makes me think, like, could we just find another formulation that's safer? If glyphosate is something that we consider relatively, not horribly safe, but we don't have another alternative, is it just like these other ingredients that are so bad? Which is something to think about, I think. But
right, and that's what I was thinking while you were talking about the ingredients, which is kind of this black hole. How do we figure out what those ingredients are? But I do imagine it would be hard to get that information.
yeah, you can probably dig and get some clue of some of the contents, but pretty sure when they have a patent like that, they don't want anybody to know what they are.
Yep. All right, so the next question, I think you kind of alluded to this, but does the type of the plant on which honeybees collect nectar and pollen affect their microbiome susceptibility to glyphosate?
I mean, I don't know about the particular type of the plant, but type of the plant in terms of whether or not it's been sprayed, yes. So, I mean, honeybees are pretty generalist. And so I'm guessing if they're foraging on plants that somebody has sprayed something around and it's drifted to those plants, or, I mean, I think that's a hard question to answer, really. I don't think anyone knows, but I would guess that it doesn't matter what type of plant. If the plant has glyphosate, that's going to increase the ability that it's going to perturb the microbiome. But if it doesn't, I don't know if that would have any effect just because it's a different type of plant.
Okay. So the next question also has two parts. And the first question is, can we get the list of studies at the end? And I can incorporate that into when I send out the Eventbrite email to let everybody know that this recording is ready for viewing. I can include the list of those studies in that email so that everybody has the opportunity to look at them if they choose. And then the second part of that question is there anything you would recommend to promote healthy gut microbiota?
So we've been trying, before I left Nancy's lab, we made a cocktail of actual gut bacteria that we isolated from bees, and we've shown that it does restore the microbiome. And we've been working on getting a patent for that so that we can, have somebody figure out how to package it and make it available for people to use. I don't know where that is right now. I'd have to ask her. So right now, I don't think there's anything available. You can, check back in with me later. We have also been testing Hive Alive, which is a prebiotic. So it's not a probiotic, but it's supposed to have things in it that help the microbes in some way and increase their abundance. So we're testing that too. I haven't seen any positive impacts on that either. So right now, I think the answer is no. We're definitely trying to work on that, trying to get something that can be used that's actually worthwhile for beekeepers to use. But that whole process of, it's mostly about the packaging, right? Because we can make these cocktails in the lab, but it's more complicated to figure out how you can make something that could be a commercial product that people could actually apply in the field, and that the microbes would still be alive, and how would it be applied. And so that's in the works for sure. So hopefully we'll have something soon. But as of right now, I can't give you any suggestions about helping the microbiome. I think limiting exposure, and then over time, they do seem to, from some of our other studies, recover over a certain amount of time. So that's basically all we can do right now until we can really get something good out.
Okay, I think this next question was posed when we were initially talking about super DFM, and Larry wants to know, what would you think about a super DFM that did contain bee microbiome organisms?
I would love that. That's what we're trying to do right now. I don't know. I mean, I guess it's kind of like a monopoly, right? Because all of us that work on the bee microbiome have all of these strains. They're available to anyone, so they could ask us for them. But it's difficult to grow them, and figuring out how to put them into, a powder or something that can be used is complicated. So I know that the makers of Super DFM have talked to me about it. They didn't seem like interested in trying to put anything real in there. But that would be ideal, right? The ideal situation is that we would have something like Super DFM, but that it has actually a cocktail of microbes that we know live in the bee and that are important for the bee, and then to be able to give that to them after, they've been exposed to any kind of perturbation.
Okay, next question. What do you think about the push to add probiotics to the honeybee's diet without having any reason to do so?
I think it's worthless. So I feel the same way about human probiotics too. If you don't have a disrupted microbiome and you're throwing new microbes at it, they're not going to colonize, because all the space is being taken up by the microbes that you already have there. So even in humans, it's been shown that probiotics don't help unless you've been given like antibiotic treatment and you've wiped out some of your microbes, and then you can take probiotics. And then in that case, it's been shown that they can be beneficial. But there's absolutely no evidence in humans or in bees or in any other animal that I know of that probiotics are worth anything if the microbiome isn't perturbed. And given that the probiotics on the market don't even have the right microbes in them, I mean, I would suggest not using them for any case right now until we have something. But then if we do have a good cocktail of the right microbes to give bees, then definitely that would be something to give when their microbiome is perturbed. But like I said, if it's not, there's no room for these things you're giving them to colonize. So they're not going to be able to stabilize. They won't be able to outcompete what's already there.
Yeah, makes sense. All right, have you or someone else done studies on the effects of RoundUp and/or atrazine on the development of larvae and the development of queens? And he goes on to say, I have noticed a tremendous reduction in queen success that coincides exactly with corn, and soybeans planting, and he's in Davie County.
So I showed a study. I can try to pull this back up a minute. The only study I know of, but I mean, there's probably more, and I can look into it. You can contact me if you want. But the one that I did find was from 2018, and they showed glyphosate is affecting larval development. That's the one I know of off the top of my head. In terms of queens, I haven't seen anything. So generally, queen studies aren't done very often because it's hard to do experiments on queens. We don't want to sacrifice all of our queens very often. But I could be wrong. There might be something that's been done on queens, but not to my knowledge. And I think we're all having, at least if I'll speak for myself, I'm having a lot of trouble with queens as well. And that's why, I'm collaborating with David Tarpey, and one of my graduate students also has a USDA grant to look at this. And so we're trying to understand at least how in-hive treatments affect queens and drones. And, maybe eventually we could extend that to environmental chemicals and things. But I think it's a very understudied area. And so we don't really know how glyphosate or other, herbicides impact the queens. Most of the work's been done on workers, of course. And I do have some tables where I collected all of the information about how each chemical that I could find impacts different aspects of workers. So if you want to contact me separately, I can try to give you some more information about it.
Okay. So it's 2:00 now, and we have 2 questions left. Do you need to go? Do we need to wrap this up, or do you have time for the last 2?
I'm fine as long as everybody else is.
Okay, we will forge on with these last 2 questions. Can you relate lab experiments as a case of what can happen to what happens in the real world? For example, has anyone done tests of glyphosate concentrations in hives, honey, or wax where the glyphosate was not directly and intentionally fed to the hive?
Yes, so there have been studies where they just sample wax, honey, and pollen from the hives, and they show the concentrations that are present there. I can't actually remember exactly what the concentrations are. They're somewhere around the same concentrations that are found in nectar and pollen when they're sampled in the environment, in areas where glyphosate has been used. But yes, there are studies that have looked at concentrations in the hive. They are present in the hive, and obviously if the bees are in close proximity to an area with glyphosate and they bring it back, those concentrations are going to continue to rise. So I think the idea for those studies, unfortunately, any time you do in a controlled experiment, you can't really put bees next to a glyphosate area, because then your control bees will have to either be in a completely different area, which could also impact things, or then your control bees are also going to be exposed. So it's a little bit complicated to do relevant exposure and have a control at the same time. But yeah, there's been investigations of concentration within the hive and areas, and they've definitely shown it's there. But like I said, I'm going to be honest, I don't remember what those concentrations were, but the 0.1, I can show you, I had an extra slide. So here, Eric was showing that from 0 to 0.1 millimolar, that's the concentration you usually find in anywhere in the environment. And then from 0.1 millimolar to 1 is the concentration that's been detected in nectar and the pollen of plants that have been recently, but not immediately exposed. And then, as I told you earlier, greater than 30 millimolar is what they apply directly to the crops. And so if I remember correctly, the amounts they find in the hive are going to be somewhere between this 0.1 and 1 area, which is what they're bringing back and storing.
Okay, the last question. Is it reasonable to hypothesize that most or all insects have bacteria in their guts, and that many or most of these bacteria would be affected by glyphosate? And then there's sort of an addition to the question parallel to the honeybees, but realizing that the non-social lives of most insects would make it really hard to design a study.
Yes, most, or actually all, I don't know of any insect that doesn't have bacteria in their gut. They all do. The difference, and I kind of alluded to this earlier from a previous question, was social insects, and that includes like ants and termites and any social insect, they have these like vertically inherited, really conserved microbiomes. When we look at things like mosquitoes and fruit flies, they do have a microbiome, and it's been shown through lots of different research that those microbes are important, but it doesn't really matter which microbe is there. So they have to have microbes, but the exact composition and what those microbes are isn't necessarily important. It's just the presence of them in general. So yeah, in that case, I'm sure glyphosate impacts their microbiome. But how that would impact health of like non-social insects that don't have a, a really co-evolved core microbiome, it would be hard to test, like you said. I think you could, maybe if you rear them all in the same diet, because then, they get their microbes directly from the diet, you could test that. But like I said, since most of them need microbes, but the microbe composition doesn't really matter, then maybe they could just be replaced by microbes that aren't susceptible to glyphosate, and they would still be okay. But I'm not really sure.
Okay, that was the last question. So I'll go ahead and wrap it up. And I'd like everyone to give Dr. Raymann a virtual round of applause for a really interesting presentation. Lots of really good information. And we're so grateful and appreciative for you taking your time today to share this information with us.
Thank you so much for inviting me. I hope you all enjoyed it. And yeah, contact me if you have follow-up questions or need anything else.
All right. Thanks so much, everybody. Have a great day.
You too. Thanks.
Bye-bye.